name is Ali Najafi. I'm a research scientist at Emery Pharma. Emery Pharma is a research and development contract laboratory based in Alameda, California. I'm very excited today to share with you a results of the method for trace-level quantitative analysis of Poloxamer in biological samples. We recently presented this work at American Society for Mass Spectrometry conference, ASMS. Currently, this method is being implemented to support a pharmacokinetic study as part of a clinical trial, as well as a release testing activities for a gene therapy-based product. Amphiphilic copolymers have been gaining significant attention due to their dual nature characteristics. So these polymers commonly comprise of two major moieties. As a result, these dual nature characteristics enable these polymers to be used for a variety of applications, including clinical, gene therapy, gene delivery, as well as being used as a therapeutic excipient. Poloxamer or poluronic is a synthetic amphiphilic copolymer based on hydrophilic polyethylene oxide block and hydrophobic polypropylene oxide block, designed in a tri-block structure. Different types of Poloxamer have a similar polymer backbone with two terminal ethylene oxide subunit and one central propylene oxide subunit. They have different molecular weights. For example, P188 contains 80 ethylene oxide subunit and 27 propylene oxide subunit, while P338 contains 141 ethylene oxide subunit and 44 hydrophobic propylene oxide subunits. Despite the wide range of application for Poloxamer, a limited number of bioanalytical techniques has been reported in literature. Here we report robust, sensitive, and specific LC-MS MRM-based method for Poloxamer quantification. Our method is mainly based on protein precipitation approach prior to LC-MS analysis. This method has been developed to address current issues and challenges associated with Poloxamer analysis in trace level. Here we show a general workflow of our method. So we generally start with the true biological sample containing Poloxamer. We vortex and add with an extraction solvent. The extraction solvent is typically an organic solvent or a mixture of organic solvents. Afterwards, we centrifuge the sample to allow protein precipitation and transfer the top layer to the new microcentrifuge tube. And then we dry the top layer and reconstitute the dry extract with reconstitution solvent. The reconstitution solvent typically has the same composition as our mobile phase used for LC-MS analysis. It is important to mention that a pre-concentration step by drying and reconstituting in a smaller volume allow us to achieve a desirable sensitivity needed for trace level analysis of Poloxamer. As part of a quantification activity, we develop an external calibration curve by spiking P188 reference standard in a true biological sample. Here we show a representative calibration curve that we obtain when we develop a method for P188 in a human serum. As you can see here, the linear range were found from 2 microgram per mil to 100 microgram per mil. Also, a low limit of quantification LLOQ and limit of detection LOD were found to be 2 microgram per mil and 1 microgram per mil respectively. It is important to mention that the most accurate results were found to be when we apply a quadratic fit to our non-zero calibrated for this method. Here we show two representative MRM chromatograms. The chromatogram on the left shows blank human serum with no spike P188. As you can see, there is no interference at the same retention time of P188. The chromatograms on the right are 3 overlaid chromatograms of 3 independent biological replicates of P188 at low limit of quantification level. So these results shows the excellent precision of the method Also, the signal to noise of P188 peak at LLOQ level allows us to do proper quantification. Also, it is important to mention that the peak shape of Poloxamer represent different components of this polymer. These components have the same MRM transition, but they are slightly different and resolved chromatographically due to different interactions with column stationary phase. We evaluate the percent recovery of Poloxamer in biological samples. So the percent recovery was calculated based on the response obtained from addition of known amount of Poloxamer to dried extract post-extraction and compared with the response obtained from true concentration of Poloxamer spike in matrix pre-extraction. As you can see here, we replicate this recovery experiment by prepping 3 independent biological samples in 3 different concentrations, low, middle, and high, all within the range of calibration curve and linear range. The percent recovery were found for all the samples from 96.9% up to 109.2%. So the accuracy of quality control samples was also evaluated. The percent accuracy was calculated based on comparing the response from spike samples to a true concentration of the samples. In all cases, in all concentration, low, middle, and high, the percent accuracy was found to be less than 15%, which is in agreement with FDA and ICH recommendation. We also evaluate the precision or repeatability of the method. The precision of the method was evaluated by prepping 3 independent biological replicates of P188 spike sample in human serum. We selected 3 different levels of quality control samples, including low, middle, high, 3.5, 35, and 70 microgram per ml, all within the linear range of the method. And we calculate the precision by obtaining percent coefficients of variation, percent CV, for all samples. And as you can see here in the table, the percent CV for all QC samples were less than 15%. We also evaluate the intermediate precision by prepping the different replicates of all QC samples in 3 different levels of concentrations at different dates, and also prepping by different analysts. And as you can see here, the percent CV for intermediate precision also found to be less than 15%. These results suggesting that the precision of the method is in agreement with the FDA and ICH recommendation as well. To conclude, we report a highly sensitive, robust, and specific LC-MS based method for Poloxamer detection in a highly complex biological sample. Here at Emery Pharma, we have a team of bioanalytical scientists who are willing to tackle hard-to-solve problems. This is only one example, but we always look forward to bringing our customized approach to every new project. Thanks for watching.